Thank you. All right, so as Yifeng mentioned, basically my goal today is to introduce you to extra things to worry about. It's not just about your own exposures or exposure of the fetus. We'll be now going several generations upstream or downstream from us and talk about how things can be passed on over many, many different generations. And just as a disclaimer, to move forward, I have to press the button back, and I'll probably get confused many times. But we'll see whether that works or not. Okay, so let's start to establish some basically background knowledge here. So maybe for you it looks like it's a really long time ago, but since the 1970s, and only really since then, we've realized that a disease could actually originate from very, very early exposures, meaning really, really early in life. And this was really epitomized very nicely by this paper here in the New England Journal of Medicine, one of the top medicine journals. And they looked at the Dutch famine cases. Have you guys heard of the Dutch famine? It's one of the very famous examples of early life exposure and later defects or disease in life. And so in this case, it actually stemmed from World War II. What happened is that during World War II, parts of the Netherlands saw complete rationing of food, ultimately actually having no access to food whatsoever because of German embargo. What ended up happening is that you had these cohorts of pregnant women who, if you look at the calorie intake here, for a small part of 1945, actually during the winter time, winter 1945, early 1946, saw really, really low amount of food. And so the fetuses inside of these women were exposed to much lower calorie intake. Then so this is the sad part of the story. The good part of the story in terms of epidemiological study was the fact that in the Netherlands, you have a really good registry, and in particular, at 19 years old, most men, like they do in Europe, have to register to the military service. So at 19 years of age, you had all these cohorts of young men who 19 years before had, in utero, had the lower calorie intake exposure. And they realized that if you are exposed, see, I did it. If you're within this cohort here, which saw for most of their pregnancy this dramatic decrease in calorie intake, then the rate of obesity in these young men, just 19 years of age, shot up by about 2 to 3-fold. So this was really interesting and actually gave rise to the blackout hypothesis and the early onset of adult disease sort of framework, which we still work with to this day. But it really highlighted that, and thalidomide, I guess, that these really early exposures could be very detrimental much later on in life, like 19 years later. But what I'm going to talk about today really extends this beyond just in utero exposure and looking at later on in life. I'm going to talk about some evidence, not in human in this case, but in animal models, that an early exposure, and by this I mean an early generational exposure, could have effects that you can monitor across many generations, including the F3 generations, which is really your great-grandchildren. So this work actually stems from the somewhat controversial work of Michael Skinner, and I'll introduce to you why it's somewhat controversial, who did experiments with a bunch of different kinds of chemicals, including the chemical vinclozolin, which is a fungicide that you use a lot to grow the vines. And so what he highlighted is if you take a pregnant mother, the mother by definition will have a fetus, and inside of the fetus you'll have the developing germline. If you expose this mouse to vinclozolin, the fetus will be exposed and the germ cells within the fetus will be exposed. So that means that the fetus is the F1, the germ cells within the fetus actually the F2, so all these generations are directly exposed. So what he did actually to complicate things a little bit more is to not look at the F1 or the F2, but actually look at the F3. The F3 is the first generation that is not represented in a pregnant mother. Does that make sense so far? And what he saw was actually effects on that third generation, so your great-grandchildren. What he showed was if you look in males at the testis, and he has looked at other organs in males and females, but the testis was one of the strongest results, you see that a lot of the cells within the testis, a lot of the sperms actually die, and this is basically here in black the vinclozolin and here in gray the control. If they don't die, the fact that they die leads to reduction in sperm number, and even if they don't die, the sperm that is left is actually not as motile, so it doesn't move as well as normal sperm. And again, that's a measure of sperm motility. It's the forward movement. You just track how much they move, and compared to control, you see this statistical difference all the way down not only to the third generation, but also the fourth generation. So extended that even a little bit beyond. I just want to show you some elements of this data. This is a control adult male exposed to the vehicle, not vinclozolin, and you can see those are seminiferous tubules. So it's inside of the testis, and you can see all these little sperms here. Those are the sperm tails that are being produced and are ready to basically make their way out. However, in a vinclozolin treated, so not that male, but again, its great-grandparent treated with vinclozolin, you see that that great-grandchild has seminiferous tubules that have all these holes and basically no or very few motile sperms or mature sperms being produced. And then he followed this work, really important work, which really made the headlines, I believe, around the world, with a series of other papers that try to track down what was really happening in these mice. And one proposed mechanism that was already floating around the literature was the fact that DNA methylation, so regulation, genes are often regulated not just by transcription factors, but by chemical groups that you can add onto the DNA. Methylation of cytosines is one of such mechanisms. And he proposed that this could be the mechanism of vinclozolin action. And he looked across the genome, and I'm showing you here some examples. So in blue are the vinclozolin-treated animals. In red are the controls. And depending on what genes you look at, you can see increased methylation, like these two loci, or decreased methylation, like this other locus right here. So it looked like not only do we have an effect on reproduction that is transgenerational, but on top of that, we also have a potential mechanism, which is by differential or aberrant methylation of the DNA. Okay, so this was sort of all nice and good until a series of controversies started to dampen a little bit the enthusiasm. One of the controversies was that the exposures that were performed, so the amount of vinclozolin that was given to the rats and mice, he worked with both species, were actually high and not considered to be relevant. Other groups tried to replicate the data in different ways and were not able to, so that's definitely a problem in scientific research. You have to be able to replicate the data. Then one of his postdoc was found to have falsified some of the data, not of these studies, but other studies, but that sort of added to some of the concerns. And importantly, some people had issue with a very biological basis for this phenomenon because of what we know happens with DNA methylation. And so I'm not going to necessarily take you through all this diagram because it's quite complicated. But to summarize what this represents, it represents the fact that at every generation, the genome erases its DNA methylation. So not just once, but twice. It happens the first time before fertilization in cells called primordial germ cells, very, very early stages of germ cell development. That's the stage where they erase almost all of their DNA methylation. It's the most comprehensive DNA demethylation that the germline will ever see. And then those cells after that regain some methylation. You have fertilization of the gametes, and then pretty soon, a few days after fertilization and the initiation of embryonic development, you have a second wave of DNA demethylation. So if indeed vinclozolin or other chemicals affected DNA methylation, how could this be passed on for so many generations down the line? Because it's supposed to be erased and reestablished, hopefully normally, at twice per generation. So there's two possibilities that people thought of, and there are more out there, but the two main ones are the fact that of all the different types of epigenetic marks, perhaps there are others that can carry out this function. So we know that DNA methylation is a very important mechanism for gene regulation, but perhaps this other mechanism here called histone modifications could also be important. As you may know, histones are proteins around which DNA wraps itself, and how you modify those proteins really influences how compact the DNA is and again, how genes are expressed. There's also a second hypothesis, which is the fact that whenever the genomes are erased, they're not completely erased. They are loci that are resistant to these waves of DNA demethylation. So perhaps it's those very few loci around the genome that retain some methylation that could be responsible for the disease. To this day, we still don't have a good answer to these two possibilities. But my goal today is to provide you with some of the evidence that we've gathered to say in other models that histones could indeed play an important role. Okay, so let me introduce you very briefly to the very complex histone code, as it's called. It's called an histone code because there are many different kinds of histones, many different kinds of modifications, and many different kinds of effects. So to summarize a lot of the knowledge, as you can see, it's kind of a complicated diagram. I really want you to focus on two kinds of marks. The active marks, and an important one is called H3K4 trimethyl. So this is histone 3, lysine 4, and you have 3 methyl groups that have been added to that lysine 4. That mark is found on the promoter of active genes. And then you have the counteracting marks to this one. You have H3K27 and H3K9 trimethyl. So again, histone 3, lysine 27 or lysine 9, and you add 3 methyl groups to it. And those marks are found at the promoters of genes that are repressed. So I'll speak a lot about these two repressive marks for the rest of my talk. H3K27 and H3K9. Okay. So how did I decide to go about this? Replicating the Skinner work was a possibility, but it's expensive. It uses a lot of animal. In his science paper, where he described that vinclozolin work, he used more than 240 animals. And to get to the third generation takes a long time. So I decided to use a different system. And what I'll show you today is to look at that effect on the epigenome. I'm deriving, or I'm leveraging, 2 different kinds of models. One is a worm, a canonical genetic model used in labs around the world called C. elegans. And the other one, if I have time, will be is a model of differentiation of the early germ cells from stem cells, embryonic stem cells. And this is done in collaboration with the Clark laboratory, Amanda Clark here at UCLA. Okay, so let's start with the C. elegans work. Why did we decide to use a worm? Well, as I said, it's a commonly and well-accepted genetic model, but there are many advantages, and the 2 top here are actually the most important one for this project. It's transparent, and it has a very short generation time. Within 4 days, 3 to 4 days, you can get another generation. So looking at things over many generations, you'll see in this talk, I'll go all the way up to the 7th generation. Doesn't take that long. It also reproduces really well, so half of the cells within the worm actually are germ cells. It can be used in high-throughput screens, and we actually understand a lot about the genes and pathways that regulate its genetics and its epigenetics. And we know how well conserved things are, and the answer is, for what we're looking at, there's a great deal of conservation of the genetic pathways. This is an example here. This is histone 3 in C. elegans at the top. This is histone 3 in human. And all these little colored boxes here basically represent different kinds of epigenetic marks. And you can see that to a vast extent, they're very well conserved between C. elegans and human. Not only the marks, but of course the machinery that puts these marks in place, all the pathways that lead to adding those marks onto the histones. And so what I boxed here are H3K4, H3K9, and H3K27, and H4K16, just marks that are very well known to regulate gene expression. Okay, so I introduced all of this, but I'm going to have to introduce yet another system that actually simplifies the way we're going to look at things. So instead of exposing the worms to a chemical and then looking at whether it's fertile and what's going on with the cells, we decided to sort of flip things almost backwards and use a sensor. So something that could tell us when it shines under UV light whether we may have an epigenetic problem or not. The way this works is we have a worm. Here this is just a section of the worm, and circled is the germline. Each little dot here within that circled area is a germline nucleus. This worm usually glows under UV. So it expresses the green fluorescent protein, GFP. It expresses GFP in every single cell in that worm, except in the germline. As you can see, you don't see any of those little dots here. And I'll show you a picture later on where you can see what happens when we actually de-repress that expression in the germline. So it turns out that, for reasons that I don't have time to get into, that transgenic worm represses GFP expression in the germline specifically through epigenetic mechanisms. So if you look at the transgene with this little dot here and here, it is devoid of repressive histone marks, and instead you actually have addition of repressive marks. So again, that H3K9 di or trimethyl, you can see that the transgene is very high in that repressive mark. So the other part that I didn't mention is that the worm is actually devoid of DNA methylation. So what that means is that the only way that this could be regulated by an environmental agent is if it acts, as far as we know, on histone modifications. So now we can take this worm, expose it to chemicals, and see whether we can de-silence the expression of GFP in the germline. If we do, then that means we're probably perturbing that nice tight regulation through histone modifications. Does that make sense so far? I see some nodding, and I'll go with it. Okay, so what do we have to do when we work with worms? We have to follow actually exactly the same protocol that I described with pregnant mice. That's because the worms are pregnant with also baby worms. So if you expose a worm to an environmental factor, you can't really look at the F1. Well, I mean, you can, but you can look at the F1 and the F2, but the true transgenerational effect will only be apparent in the third generation. So again, that great-grandchild sort of paradigm at the level of a worm. But the advantage of using a worm, as I told you, is that to get to this generation, it only takes 2 weeks, not almost a year if you're going to do this in mouse. So we can get very fast sensors in the worm. And because we're using that sensor, what we're trying to see is if we expose that worm to a chemical, will we see desilencing of that GFP report in the germline? And if so, does this get inherited across generations? I'm not going to spend a lot of time on this. We validated the use of this reporter by using a bunch of drugs that we know affect histone modifications. So these drugs have been used by the pharma industry, by a lot of researchers around the world. We know exactly what they do. They target different kinds of enzymes that add repressive marks or remove repressive marks, add active marks or remove active marks. And basically, whenever we added active marks or removed repressive marks, we saw this desilencing of the GFP in the germline. And you can see now all these little dots here, all these little single germline nuclei that show up by expressing GFP. So it's something that is fairly easy to see under the microscope. So maybe that's going to be a surprise, but we actually did not use vinclozolin for our studies. There are several reasons to this. One of it is that we, as a crapshoot experiment, we just decided to expose the worms to BPA and ended up seeing actually a very resilient effect. I had done some work with BPA. Other people in my lab were working with BPA. So we thought, why don't we just expose the worms to BPA and see what happens? And we actually saw a very strong effect. If you're not familiar with BPA or bisphenol A, It's a component of plastics that is widely used as a plastic manufacturing chemical. You find it in plastic bottles, in the liners of food cans. It's not just aluminum, food and soda cans, and it's not just for cats. It's basically all cans. You find it in dental composite, and the biggest worry right now is thermal paper that has really high levels of it in the micrograms, and it looks like it can get through your skin pretty easily. It was also a good choice because we know that BPA exposure can affect reproduction, and we know through other people's work that adult germ cells, so later stages of germ cell development, the epigenome of the cells is sensitive to BPA exposure. So we thought, let's just give it a try. So I'm going to show you now different diagrams, and here at the bottom you have different generations. So again, this is the parent exposed to the chemical directly, then that F1, F2, and then that great-grandchild generation, the F3. If you expose the worms, that sensor, to water, we see nothing. We see that about 90% of the worms show really nice silencing of the GFP in the germline. If we expose the worms to DMSO, which is the solvent that unfortunately we need to use, not just for BPA but for a lot of other chemicals, we see unfortunately that little spike. Oops, go back. That little spike in the F1, where you see some desilencing, but it goes back down, not completely back down to the water level, but significantly, so that by the F3 we still have a small difference with water, but it's nothing close to what we see with BPA. If we expose the worms to BPA, they start by desilencing a lot more, about twice the amount of desilencing that we see in the DMSO control, but then it shoots up to about 50% and plateaus at around 50% all through the F1, F2, and third generation. So, and this is highly statistically significant compared to DMSO and of course water as well. So we do have an effect that is transdurational. Now, this seems quite artificial because it's through the lens of this reporter that expresses GFP, and so what, basically, for the worm, or even for us. Oh, sorry. Before I get into that, we tracked things recently beyond the F3, and we see that this effect actually lasts and only resolves once you get to the 6th and 7th generation. So it's actually a really long-lasting kind of effect. Okay, so as I said, that was through the lens of a reporter, but if we look at the fertility function of those worms, so how well they reproduce, and we can do this easily in the worm by monitoring how healthy the embryos are, so how often they die. That's the rate of embryonic lethality. We can see that by the 3rd generation, there's still a much higher level of embryonic lethality of BPA-treated animals compared to the DMSO control. So I'm representing everything as a box plot that allows you to see really the breadth of the data. It may not look dramatic in results because I'm plotting everything, but this is highly statistically significant. So not only do we see a transdurational effect through our reporter, but we also see a transdurational effect with regards to just looking at the fertility of those worms. These worms are less fertile. Okay, so this is my sorting hat. I am a very big Harry Potter fan. Very excited about this Saturday. And the next set of experiment is all about sorting and segregating populations. And so it's not just a sorting hat, it's a thinking hat on. There's going to be a lot of different directions for the data. So stay with me for a second. So what we did in this series of experiments is we took those worms and we looked at how well they responded to BPA. If you expose them, if you take those parents and you expose them to BPA and they don't respond, you see no desilencing in the germline of the reporter, then we put them in a separate bin, a separate set of plates, and then followed what was happening all the way down to the grandchildren. However, if another group of worms seemed to respond really highly to BPA and we saw desilencing of the germline, then we took those worms that responded, again through the lens of a reporter, and then followed what was going to happen to their specific progeny all the way down to their great-grandchildren. And that was to try to see if the worms that respond as parents have a disproportionate result in their children. Okay, so this is what the data said, and basically the bottom line is it's confusing. So if you are a parent exposed to BPA and or DMSO as control, of course, and you don't show any response, then with DMSO you're going to go back up to that sort of plateau at around 20%, a little bit higher than water. But if you've been exposed to BPA, you also increase in the F1, but you continue increasing, so that by the F2 and the F3 you actually have a response that is statistically significant compared to the DMSO control. Now this is very different from the opposite, I guess, result, or the other half of the experiment. If you are a parent and you respond strongly to BPA or DMSO, then what happens to your progeny? In the case of DMSO, it actually shoots back down, so you have 100% of the parents responding, showing desilencing because we selected them. But then in the F1, in the F2, in the F3, it goes back down, and you have nothing, something that is not significant compared to the other DMSO. However, if you've been exposed to BPA, you go back down in the F1, but then you plateau and you stay at around 60%. Okay, so let me summarize all this. What this means is that whether you respond or you don't, you're still going to see something different from control. There is a difference between worms that responded as parents and worms that did not respond as parents. There's a small difference, and it's statistically significant, but they're still each of them different from the control. So you can't really predict, or not very well, what will happen to your great-grandchildren based on your own response. That's the bottom line. If I respond or if I don't respond, that has kind of little consequence for my grandchild. All that matters is that initial BPA exposure. Does that make sense? I saw some nods. I'm going to go with that. Okay, so then what is the mechanism behind all of this? This is where we could use the worm and the genetic tractability. That's why it's such a good model system. So we could take these worms where we saw a really high rate of, no, the other way. These worms here that show this really nice plateauing of the effect. It's also the strongest desilencing that we see. It's 60% and it plateaus. So it's really high, nice heritability of the effect. And we can try to intervene in the middle of the generations. And we can intervene in different ways. We can intervene by exposing the worms to chemicals or by exposing the worms to RNA interference, which is a way to modulate gene expression. Okay, so first I'm going to show you what we did with chemicals and what I mean by chemicals. What I meant by this is actually using a combination of drugs, again, very well characterized drugs used in the pharma industry, that we know will lead to an increase in the levels of repressive marks, H3K9 and H3K27 trimethyl. Okay, so these drugs should actually make the chromatin, the DNA, more repressive. And if you look at the gray here, which is those drug-treated worms in the generation prior, we see that the rate of desilencing is much lower compared to the worms that were exposed to BPA alone. So in gray, is BPA and IOEX1 and GSKGA4, or in this case, it's those drugs plus DMSO, right? So in both cases, we see that decrease that tells us that those repressive marks are important for the inheritance of the desilencing phenotype across generations. And it's not just a question of reporter. We also see that the embryonic lethality is reduced, not dramatically, but significantly reduced when we treat those worms with these drugs. So that meant that indeed those repressive marks are really important for the desilencing of the reporter and for the embryonic lethality that we captured. Okay, but this was doing this chemical rescue of the phenotype, but we could try to get perhaps more granularity on what was going on by doing a genetic rescue. So again, this is doing RNAi, RNA interference. This allows us to downregulate the expression of genes. And to make a long story short, We looked at different kinds of enzymes. Specifically, we looked at lysine demethylases that either target K9 trimethyl or K27 trimethyl. So just like what we did with the chemical rescue, if you downregulate these genes, because they are demethylases, you should increase the level of these marks, these repressive marks. Does that make sense? So just remember, when we do here this GMGD2 RNAi or this combination RNAi here, what we're doing really in practice is upregulating, increasing the level of these repressive marks. And in both cases, we see this really nice, compared to the control here, we see this really nice downregulation of GFP expression in the third generation. So again, that meant that the regulation of these two marks is really important for that phenotype to become transgenerational. Again, it's not just through the lens of a reporter. If we look at embryonic lethality, the strongest results that we got actually was with these enzymes here that target K27 trimethyl. In this kind of RNAi, we saw this really dramatic dampening of embryonic lethality. So if you really upregulate that repressive mark, H3K27, you don't see a transgenerational effect. OK, so what I've told you so far, let's see if I have time to get through some of the other set of data. But what I've told you so far is that BPA does cause a transgenerational effect, at least in C. elegans. It's still to be proven in mammals. And that this transgenerational effect is caused by the deregulation, likely caused by the deregulation of repressive histone marks. One set of data that I've not shown you yet that we're still working on is actually looking at the DNA and looking at where those histone marks are and whether they are indeed affected by BPA. And as I said, we're still working on this. But for the last few minutes, I want to switch gears a little bit and tell you about a different model that hopefully will complement our C. elegans data. And that's a model of germ cells generated in vitro from embryonic stem cells. These are all done in the mouse. And mouse ES cells are cells that you can commonly grow in the lab. But thanks to people like Amanda Clark here on campus, now you can take these cells and make aggregates of cells that will contain germ cells inside of them. And what's really interesting about these germ cells that you make in vitro is that they capture that exact stage where you should see complete remodeling of the epigenome, including removal of DNA methylation, but also addition and removal. There's also quite an extensive remodeling of histone modifications. So we can hopefully look at these guys in differentiating cells. I just want to show you what these look like. This is sort of a quadrant of this aggregate. You have a bunch of different cells inside of this aggregate, but we actually have fluorescent markers. So here you have the blue marker and the green marker. And when they overlap, like these two cells here, you basically have a germ cell. And this is one of the repressive marks that I mentioned before, H3K27. And H3K27 actually increases in those cells as they differentiate. So now we can look at what BPA does. This is just a close-up of the cells. So we can expose those cells to different kinds of chemicals. We can do this for several days. We can look at the survival, how well they differentiate, and of course we can look at the levels of epigenetic marks. This is basically how we look at the cells. We can look at them under immunofluorescence, which was what I was showing here. This is immunofluorescence. But you can also just take those fluorescent cells and put them through a cell sorter that will detect the levels, the presence of the cells and the level of fluorescence. And here, this is basically, this quadrant here represents the cells that we care about. Those are the primordial germ cells. And those are the cells before they differentiate. And of course, you don't find germ cells before they differentiate themselves. And through a beautiful work from my postdoc, what he showed is that we had a window of sensitivity here. So what does that mean? That means that before you have germ cells, when they're still what are called epiblast-like cells, so they haven't differentiated into germ cells yet, and you expose them to a range of concentration of BPA, they do actually fairly okay. In some cases, we see even more viability of the cells at lower BPA concentrations, even compared to control. So before they differentiate, these cells are actually perfectly fine with reasonable amounts of BPA. However, as soon as you are able to differentiate into PGCLCs and you expose them to the same BPA concentration, you can see that at almost, well, actually at all concentrations observed, we saw a decrease in the number of these cells compared to control. That meant two things. That meant we don't differentiate them well, or perhaps they die when they differentiate. We don't know actually yet what is really happening here. But the bottom line is that primordial germ cells show sensitivity, but the cells right before that stage do not show that sensitivity. So that stage of germ cell development is really a critical window for the exposure to chemicals, at least to BPA. And we're testing other chemicals as well. So we see that decrease in germ cell number at all concentrations. We're now testing lower concentrations. But if you look at the surviving cells, we also see at low intermediate concentrations reduced levels of, again, the two repressive marks that I told you were really important from our worm work. We saw decreased levels of H3K9, and we also saw decreased level of H3K27 trimethyl. Of course, because a lot of the cells died, we don't know what happened to the cells that died. So perhaps these cells, perhaps if you look a little bit earlier, when you have more cells still alive, you would see that even lower levels of H3K9. Perhaps the cells that had very low levels died because these marks are important. Okay, so I told you a lot of stuff today, but let me summarize it. From the worm work, what I showed you is that BPA exposure causes a heritable germline chromatin desilencing effect. So we have this beautiful sensor. It's really clear. You express GFP or you don't. You expose the worms to BPA, and we see this chromatin desilencing effect. Desilencing the chromatin of germ cell is never a good thing. You want to keep it tight to make sure that the genome remains stable. And of course, if you desilence the germline, then we can see that indeed the germ cells and the embryos that are generated from those germ cells don't do well. And we also are able to capture this embryonic lethality phenotype that is also transgenerational. And the body of work that we've done and that showed through chemical rescue and genetic rescue also told us that a lot of these phenotypes are dependent on the proper regulation of two types of histone marks, repressive histone marks, H3K9 and H3K27 trimethyl. So we mirrored at least some of this work through a in vitro mouse primordial germ cell model. We, at least some of the work, I should probably suggest we need to repeat it and do different kinds of windows, but we believe that that time of early germ cell differentiation represents a critical window of sensitivity of these cells to environmental exposures. And again, that BPA seemed to be regulating the expression of K27 and K9, especially lowering the level of these marks. So we're really excited about that primordial germ cell-like cell model because it will really allow us to interrogate what is going on perhaps in a more human-relevant context at the level of the chromatin, and really interrogate these cells as they're undergoing that very critical remodeling of their genome, epigenome. So I really want to thank the people who have been doing all this work. So these are the members of my lab, but the people directly involved are these three here. Jessica actually started this whole project. Lisa has recently joined and has been really crucial for doing a lot of the genetic rescue that I showed. And then Danny is responsible for doing the primordial germ cell-like cell, so the mouse work that I showed at the end. And I should have had Amanda Clark here, who's been also instrumental in helping us import her model into our lab. And those are my funding sources, and I'll be happy to take any questions for the next 5 minutes or so. Thanks. Sure.
Is yours the only model verification now? Or have other people verified the initial model as well?
Okay, so we have not, so if I call myself a model organism person, small model organism person, we've not necessarily all converged towards the same kind of endpoints and the same kind of chemicals. So some people in zebrafish, for example, showed TCDD transgenerational effect. That's fine. I showed that in the worms. People have, forget what they've done, but in the fly it also applies as well. So it looks like any time that you work with smaller model organisms, you can really easily detect those transgenerational effects. Maybe not easily, but we can detect them. But as soon as you look into the mouse or the rat, then this is where the data becomes messier and things just don't seem to, as far as people have looked, we haven't had a very strong study beyond Michael Skinner's one, which again was controversial. We haven't had something really strong saying, yes, it's real, we should believe in that. So hopefully what we're doing will establish a foundation for doing targeted studies in mammalian systems so that people can know where to look, what to look for across generations.
You verified Skinner's original model, but you're developing on a simplified basis.
Exactly.
To that model.
I'm breaking it down into smaller elements. I'm breaking it down by using a Cialingus model, and I'm also breaking it down by just looking at that window where if we see that these marks, these changes get erased, then they shouldn't be transgenerational. If we see that they last, then there's a clear cause for concern and transgenerational effect. But if the early mammalian germ cells seem to reset all these marks, then it's unlikely to be legit. Yeah?
Why is it that we only see these changes in the third generation?
No, so I sort of skipped the middle generations, but we actually do see desilencing and some level of embryonic lethality in the intervening generations. The problem is that we compare everything to DMSO, and DMSO in F1 also has quite a bit of an effect. So it's really by the third generation that we can see the most difference with DMSO. And again, it's not just the third. We also see an effect in the fourth and the fifth. And by the sixth, it goes back down to the DMSO level. Does that make sense? Yeah? No, you do the RNAi just like you do. So the rescue that we did was actually in the middle generations. So what we did is make sure that the worms responded, that they showed the effect, and then we wait for one generation, then we do RNAi, and then we look in the third generation. So we...
RNAi was not done at the third generation when we were looking?
No. Right. So what we wanted to do was not to prevent the effect in the first place. We wanted the effect to take place and then see what could block this effect from being inherited across generations. But the other, what you're suggesting, or what you thought I did, I guess, is also an important response. What is, it's a slightly different question. There's what is necessary for the inheritance, and then there's what is necessary for the initial response. They might be different, they might be the same, we don't know yet. Any other questions? All right? Oh, okay. I was going to wrap up.
The slide that you're showing where the parents have 0 response versus 110% response, and their offspring sort of comes towards in the middle.
Yeah.
It seems very, like, very classic, central tendency type of phenomena that you would expect where because your artificial is let down 0 respondents and 100% respondents, you would expect the next stage will,
so this guy, right?
Towards the central tendency.
So I think what you're alluding to here is what would happen if I gave that data to a statistician? Is that what you're suggesting? Yeah. So I mean, obviously we've done stats, and we think we've done stats the right way here, but it would be interesting to perhaps extract more statistical explorations of this, including trends and things like that.
The artificial is like 0 and 100, right? But you have parents that's between.
Right. So I'm not necessarily puzzled by this decrease here because it looks like the system is stabilizing somehow. But what I was puzzled with by was this guy. this trend towards more and more response as generations keep going when you selected the worms that did not respond initially. And you know, in future work, perhaps we want to see whether this trend keeps going up or how quickly it goes back down or things like that. But that, to me, was the puzzling part, that even if you don't respond as a parent, that doesn't mean your great-grandchildren won't be safe, unfortunately. Again, more things to worry about. All right, thank you.